Today I wanted to demonstrate some of the uses of Megaline Pro, a little bit on how the software works, and also how to create an alignment, but also some of the tools that an alignment can be used for. Sometimes the alignment itself is not the end goal. You're working towards something further. All right, and before I begin with Megaline Pro, I just wanted to say a little bit about DNASTAR itself. So it's a life sciences software company. It's been around since 1984. This is the oldest life sciences company that hasn't changed hands in that interim. Originally, the software included molecular biology applications, but over the decades has expanded to include structural biology applications, working with proteins, and then somewhat later, genomics applications, which would be your next-gen analyses applications. And Lasergene is the most cited life science software. Now, I thought it would be really interesting to show a timeline of some of the great moments of science where Lasergene software was used in the analyses, but I found this to be really difficult because there are so many Lasergene citations out there that it just became subjective as to what the great moments of science are. So I just chose about 5 really interesting ones just to hit upon. So here's a picture of Utsi. He's a Copper Age man who lived and died about 5,000 years ago. His body was recovered from a glacier in northern Italy, and his entire mitochondrial genome was sequenced, assisted by the use of Lasergene software. And he was also, the populace at large was surveyed, and they found that people are still genetically similar to certain markers with his genome. They live in southern Italy today. NASA has used Lasergene software. They collected bacteria from Mars and did a phylogenetic diversity study on this. All the bacteria they found originated from Earth. Lasergene, by several researchers, is being used to this day to study bird flu across the world. Lasergene software was used to study the genealogy of the Tutankhamun family, as well as identify several of the pathogens that the people were afflicted with. Modern science is definitely a good thing. And just another use of Lasergene, it was used to create an assay for jewelry and other ornamentations that are made out of bone to identify whether the bones are being harvested from endangered creatures, and in some cases they even identified extinct creatures. The bones are being used for jewelry. So Megaline Pro, this application is primarily designed for creating alignments. It can do multiple sequence alignments. It's capable of handling genes and all the way up through the genome level. It can handle RNA, DNA, amino acid bases. And the alignments can be built upon. You can either merge, combine different alignments into one alignment, or you can have an alignment and continue to add sequences over time, making an ongoing project. All right, so moving into our molecular biology suite. So here I'm going to transition to Megaline Pro itself. I'm going to start with the DNASTAR Navigator. This is an interface that's included with the Lasergene software. It's a great resource for accessing all the applications without having to root through files. Here there's the categories, molecular biology, and we also have additional categories below, which aren't the focus of today's talk. So I'm going to move to the Megaline Pro tab right here. So if I click this, it'll open the application, but before I do that, I just wanted to point out if I hover over this application for a little bit, there's a descriptor of the software that pops up, tells you what it's for if you're not familiar. And in addition, there's resources below. I can quick link to different videos, tutorials on how to get started. I'm sorry, these are videos. Below that are tutorials. And if you're looking for additional resources on any of these applications, the DNASTAR website has additional recordings, tutorials, and also has pre-recorded webinars available. So I'm going to click on this link and open the software. And here's the interface. So the first thing I'm going to do is just walk through an alignment. So I'm going to move up to File. I'm going to add sequences. These are sequences I gathered from GenBank. I just did a keyword search using Neanderthal. I'm sorry, I didn't say what I did here. Here I'm left-clicking on the top sequence of interest and shift-clicking on the lower sequence. And I can bring them all in at once. And I mentioned, actually, let me say a little bit about the panes here. So there's two main windows here. There's an upper one. I'm going to lower this view just a little bit so we can see it better. There's an upper view here called the overview. And these are just a representation of the sequences that were imported. There's a scale on top. I can see roughly how long those sequences are. And down here in the lower right, there's a little data generated from what I imported. 13 sequences. They're all roughly about 16.5 kilobases. All right. I'd like to show some more about the data, or I'm sorry, show some more about the features of the interface, but I actually need an alignment to work on first. So what I'm going to do is if I move up to alignment, I have a series, there's 4 algorithms that can be used to create alignments. The upper 3, Clustol, Omega, MAFT, and Muscle, these are general use alignment algorithms. It's a matter of personal preference, which one you prefer. Over the years, I've just kind of moved toward Muscle myself. It's a highly cited algorithm. It's cited at least 100,000 times. And then the last algorithm, MAUVE, I'll cover at the end of this talk. That's meant for using whole genome alignments. All right. Now here I would click on Muscle. However, this is going to take about 2 minutes to align, so I'm just going to move to that alignment already underway. Here I haven't altered anything. All right. So I had mentioned earlier, well, before I continue, I just want to say the interface itself for MegAlign Pro has a lot of customizability. There's a lot of control at your hands to make things just how you want them, for either just as a matter of preference or as using it as a tool to help scan your data easily. So the first thing I want to point out is these were sequences imported from GenBank. It's automatically given these sequences the name, the GenBank accession. I want to change these to something a little more meaningful. Here I'm opening this thing up just to see all my sequences. I'm going to left click on the top one, shift click on the lower one. What I want to do is I want to change the name. So here I right click on any one of these sequences that is highlighted. I'm going to go to rename of fields. So these are the fields brought in with the accessions. I'm going to remove that, and I'm going to bring in what the organism is and some kind of description so I can actually tell the different organisms apart. Clicking OK. So here I have my sequences. I have the name. These are all Neanderthal sequences and a little bit about where they were collected, the provenience. There's one I brought in that is not Neanderthal. It's a modern human sample. I'm just going to show you a different way to rename the samples. Other than using the fields that are given with the file, you can give them your own name. There's a really long name. Let's just change that to human. All right, so you have the ability to change the names of the samples. There's other ways we can change the interface as well. If you look over here on the right, there's a Style tab, a Tracks tab, and a Features tab. I mentioned that these are accessions brought in through GenBank. I'm going to open this up by clicking this little tab. I can see that there's features that are still included with the information. If I slide the scale, I can make that just a little bigger so we can see it better. Okay, these two windows are synchronized. If I open up the lower samples as well, these same features are in place. And there's this shield-shaped structure. This is this blue line coming down. That's actually a reflection of what's visible in the lower sequence pane. So if I move this over, say to right about there, this blue line is reflected in the lower window, and I can see the same features that are represented above. Okay, the reason I wanted to point this out is because if I move over to this Features tab, I can actually control which features are being shown. If I'm not interested in these different things, I can turn them on or off. I'm not sure if you noticed this. The features were, as they were being turned off in both windows at the same time. The windows are synchronized. And just to further drive that home, like if I want to bring this human sample to the top, I'm going to drag them to the top, and the overview has changed accordingly. Okay, so moving back over to these tabs. So I showed you the Features tab. For the purposes of this webinar, I'm not going to focus on the features. If I move to the Tracks tab, these are any of the non-data related things that you'd see, whether the scales or the different resources to use, such as the sequence logo below. I can control what I'm turning on, what I'm turning off. So maybe I'm not interested in this consensus. I can just turn that off by unclicking the box. There's two rulers in place here. There's one at the bottom. It's off screen. I'm just going to turn that off also, just to give myself slightly more room. And there's some other interesting tools too, such as consensus match. I'm going to click that on. It pulls up a histogram. Now this particular data set is very similar, highly conserved, so the histogram isn't really the greatest resource in this for this data set. I'm trying to find a place of variability here. Anywhere where there's a difference, there'll be a dip in the histogram. If you have a less conserved data set, this actually becomes easier to see. But you can get the idea right here. I'm going to turn that off. And the last tab on the right is the Styles tab. This is more controlling the data itself, what you're working with. So in the upper overview pane, there's a window right here. I can actually control what I'm seeing here. I can change the fonts. I can change the spacing between the sequences. Change the size, even the color, I believe. Moving down to the sequence view, which is primarily the area where I'm interested in. I can change the layout. It's not going to make a difference at this point because my sequence is filling the whole screen. I can change the fonts, the colors. I can change the color of the chemistry if I want. I actually prefer the default setting myself, so I'm going to go back to that. And say I wanted to use this information here for some purpose. So for instance, I have this one human sequence in there, modern human. If I wanted to compare the rest of the Neanderthal sequences to the human to identify some positions that the Neanderthals have that the humans do not, like a character state that's different among all the Neanderthals, I can do a really quick comparison of the data. So if we move over to comparison. So I want to compare something. How about show only differences from the reference? And I move the consensus, so I'm going to have to reset that to the human, which is what I want my comparison to be. So here I can rapidly identify positions that are uniform throughout the Neanderthals and different from the human. And I can quickly click through these and I can identify them very rapidly. Had a couple back there. Here's another one, another one. So using these comparison features, depending on what you're doing, if you're looking for differences or certain whatever you're looking for. There's ways to make them visually more outstanding, make them pop to identify. All right, there's a couple things I haven't shown yet with this interface. There's some new tabs that arrived that showed up once I completed the alignment. There's a distance tab. So a distance matrix was created for the samples automatically. There's also some controls on that. For instance, this data is uncorrected pairwise. I can change that to mirror knee if I want. Easily changes back. And also a phylogenetic tree is generated. This is done automatically. I also have some controls for this as well. I can change the layout. Actually prefer this view. Change the fonts, the background color. I can change the color of the branches themselves if I want. I change it to a dark blue. I can change the width. So there's some ability to change the interface here, change the view. And sometimes people want this phylogeny for either a publication or a presentation or perhaps even a class. If we move up to File, I can export the image. Here, I can export this tree. It can be exported as a PDF or JPEG. There we go. So that's usable that way. You can export it. Similarly, I showed you can export the tree. You can also export the sequences or the overview if there was something you wanted to show in that. The data can be exported as well, either sequences, alignment, different settings there, distant matrix tree file. And something else I want to show. So I have about 20 years experience in phylogenetics. So I tend to make trees that are the final product, and I need to put lines in there or highlight by color or do some enhancements that I need other software to do, fine enhancements. Here I've set kind of like a rapid link to pass my tree file off to Figtree. Figtree is just a free software, has some enhancements I can do with the final tree to make it more, I guess, some final enhancements that I'm looking for. I'm not going to click on that. And one last thing I would say is if I wanted to put in lines or put some kind of emphasis on this tree, one of the softwares I found that works really well for that is Adobe Illustrator. And when I showed the exporting the tree image, exported as a PDF, you can import that PDF right into Adobe Illustrator. All right, having said all this, I'm going to move on to showing you something else. Let's go to a new file. And I'm going to show you a different way of importing data. So the first time I did it, I went up here, added sequences through the File menu. Let's back out of that. I'm just going to open up a data file, and I'm going to drop in a FASTA file, or FASTA, depending on how you pronounce it. And I just dragged that and dropped it right on here. It's going to prompt you, what is this kind of data? It is DNA in this case. And just to briefly tell you what you're looking at. So I'm going to use this use the software as a tool at this point. And what I have here is I'm a plant phylogeneticist. I tended to work a lot with polyploid plants. So these are clones of a particular nuclear gene from a single plant. It is an allopolyploid plant, so you have what is effectively multiple species hybridizing together to form this plant. So you end up with the genomes of different species brought into the single genome of this plant. So when I was looking at this kind of research, I didn't have any sequences to base what I was looking at off of. So what I was doing is I was cloning this PCR reaction and trying to identify the different donor species genome versions So generally, I would have about 50 to 200 of these clones. Here, I just have 3 for simplicity's sake. I'm going to move up to align. Just click muscle. I intentionally chose 3 just so it's very rapid. And what I was doing, granted I had a lot more of these, I would be able to go through and I'd be able to see motifs that were characterized among the different clone sequences. Here, clone number 8 looks kind of different, so I'm just going to move that off to the side to group these lower 2. Now, only looking at 3 sequences, it's hard to say whether these are allelic versions, these lower 2. There's some really minor differences. Here, there's a C where there isn't. There's a couple more differences. So these could either be some kind of PCR miscalling by the ABI software that identified the trace files. But you can see that that upper sequence is much different than the others. And it ended up, I used this exact method to identify the different progenitor genomes that existed in polyploids. I had to start somewhere, and this is the method I used. And I find the alignment worked really well just to get a bearing on it, an idea, get a count. This particular sample was an octoploid, and once I identified 4 distinct versions of the amplicon, I was able to, with some confidence, believe those were the different homologous reads, the different versions of the gene. All right, one thing I want to point out before I move on is here there's a position in this alignment that isn't exactly how I would align it. To me, it doesn't quite look homologous. I want this A to be over here with this column of A's. And one of the great things about this software is I have some control over that. So what I would need to do to move this A in line with the rest is I need to introduce a second gap on this other side. So the way the algorithm is used, I'm going to move up to align. Instead of hitting muscle again and using the default values, I'm going to move down to align with options. All right, so it shows me the different parameters, and at my control, I can change these. So I said that I want to add a gap to make this alignment a little more homologous, at least to my eye. I can reduce the gap penalty. And granted, you may not know what all these controls do. But there's not a lot of them, and even with trial and error, where you can read about them and figure out what they do. Here I lowered the gap penalty just a little bit from -400 to -300. I can click Align, it's right there. And it's realigned my sequence. So that position of interest was at around 300. There we go. So I can see the A's have come together in a column, and I have my 2 my 2 gaps introduced. And if I move through this sequence, granted there's only 3, but everywhere I look, this is exactly how I would align it were I aligning it by hand. And the reason I point this out, maybe you think I'm harping on this a little too much, is last time I published, I did an alignment by hand, not using this software. And the editor and one of the reviewers were asking me to quantify and qualify all the changes I had done. And this is pretty much impossible to do if you've made subjective calls by hand. So one of the great things about this software is you can just say, I use Megaline Pro version 14. I used the muscle alignment method, and the parameters were all default except I had changed the gap open penalty to -300. That would actually save me quite a bit of time trying to explain what I had done. All right, I'm going to move on now to MAUVE. And I've already loaded this because these are some quite large sequences, and it took maybe about 20, 30 seconds but that's longer than I wanted to make you wait. I just resized that window slightly by dragging our little grip bar right here. So I have 6 bacterial full genomes I've loaded in, and the sequence view is visible down here. And I just wanted to show you this in contrast. I've already done the alignment as well, and the alignment took about 3 or 4 minutes. Again, I don't want to make you wait that long. So here I have my full genomes. I can see the size down here in the corner, 1.5 to 1.7 megabases. And what I did is I went to Align and clicked on Mauve and generated this view. All right, again, I'm going to pull this down just to see the upper view a little bit more. I can grab this little spyglass thing to resize. So this is the alignment, and these are not features that are described in the data. These blocks, like for instance the green blocks, the Mauve analysis goes through and looks for homologous regions and identifies them and tags them with a color. And it allows you to align whole bacterial genomes. And the importance of this is when you're looking at entire genomes, frequently you get rearrangements, which renders other methods of alignment incapable of completing them. Once you start moving regions around, other methods just will not work at all. They end up spacing them and having large gaps. Nothing ends up being aligned except for small portions. So here I have all these little lines are tracking the different homologous regions across the chromosomes. And if I'm interested in a different region, say this, I don't know, this green one here, I can double click on it and it rearranges the alignment below to reflect what I've clicked on. And notice the portions that are outside of the alignment are going to have this kind of purplish gray or hue to indicate they're not part of the alignment. The alignment itself is still in those original colors as I had dictated, for instance, in the color chemistry. And if I move off to the other side, they become that purplish gray color again. And I'm trying to find one that, let's see, if I click on a couple of these. There, that was an example of what I was looking for. If you notice this sequence right here, and I believe one below it, let me lower this down a little bit. These two sequences, they actually had to reverse complement themselves to fit into that alignment. So the Mauve analysis will automatically look for that and do that for you. All right, so that's the Mauve analysis. Great for doing whole genome alignments. Oh, one last thing I wanted to point out about that. The phylogenetic tree for these regions that are rearranging will be different for each section because there's a slightly different evolutionary history among each of these portions of the genome. Generally, the phylogeny won't be very much different, but it's capable of having a slight difference. So it'll be rewritten in different cases. And what some people do is they gather up these phylogenies and they can overlay them and look at the relationships among the different regions and use that as their data. All right. That concludes my webinar. Just a moment, please.
You're not quite done yet.
Not quite yet.
We have several questions. I've answered some of the questions via chat today, and we're going to answer some of the longer questions. I'll just have you email those participants afterwards. But I did save 3 of the questions that are quite quick to answer since we're right at the end of our 30 minutes. So these are just pretty quick ones. And the first one is, what is the algorithm behind building the phylogenetic tree? Is there any way to tell what algorithm was used?
Oh, yes. So the algorithm that's automatically, oh, sorry, microphone. So yes, the algorithm behind the phylogenetic tree is neighbor joining. And there's a little indicator right here. I don't know if it's the clearest. It says BioNJ. The NJ is neighbor joining.
Great. And the next question is there a way to make the bottom half of the screen larger if I don't want to look at that top portion, that overview portion?
Yes, there is. So if you're primarily interested in the sequence view, where I'm circling, and not necessarily in this upper view, the overview, there's a couple different things you can do. Here I've moved down to this little corner. There's a, I'm going to click on it again. I can return back. I can toggle on and off. Remove the split view. It's essentially removing the upper pane temporarily, just kind of like minimizing it. And if you want to see more of the sequence all in one view, this is a good example where my sequences aren't taking up the entire screen. I can move to, say, automatic wrapping, and then it's continuing on the other side. I can look at multiple lines at a time. If I wanted to go up to tracks, I can even remove the sequence logo and the rulers and things like that to even view more of the sequence at any given moment.
Thank you, Brian. And one last question is, how do I know which of those 4 alignment methods are best for my project?
Okay. Yeah, right. So there's 4 alignment methods here. MAUV, definitely you want to use if you're working on a whole genome, say for a bacteria. And these upper 3 are a matter of choice. If you're interested in learning more about the individual alignments, algorithms themselves, even their history, you can move over to the help column, or the help feature. And I'm just going to go to index and look at things in broad categories. Let's see. Choose the multiple alignment method. Here, I'm going to click on this. Oh, here we go. Yeah, here's a description of the 4 different algorithms. It's showing where they were developed, location, the publications they originally citing them, and a little bit about each of them, which I'm not going to go into detail at the moment. And there's also additional links here where I can click on them and move into more detail about them. Multiple alignment algorithms. And here's even the alignment-specific links I could click.